let's bring on our first speaker today, Snehal Patel. He's the CEO and board member. Wait, there he is, of board member of Greenwich LifeSciences, ticker GLSI. There he is, sir. How are you doing on this Wednesday morning?
I'm doing very well. Thank you, Spencer.
Great. Excited to hear from you. Excited to hear about the company. Let's go ahead and guys, let's bring up his slides. There they are. I'm going to hop off the screen, sir, and then the floor will be yours.
Okay, great. My name is Snehal Patel. I'm the CEO of Greenwich LifeSciences. Thank you very much for joining me this morning. This is our safe harbor. So I'll give you a summary of Greenwich LifeSciences. So we're planning a phase 3 trial for GP2. It's a 9-amino acid peptide of the HER2 protein. We mix it with GM-CSF and inject it intradermally. We treat patients, breast cancer patients following surgery who are HER2 positive, HLA2. And following surgery, they get Herceptin or Kadcyla, and then they receive our treatment. And our phase 3 trial is designed based on our phase 2 trial, where we've seen no recurrences over 5 years if the patient was fully immunized. And that's versus an 11% placebo recurrence rate. So it's 100% disease-free survival versus 89% disease-free survival in the placebo, or 0% recurrences in the treated versus 11% in the placebo. And that data is statistically significant. We've shown peak immunity at 6 months, minimal to no side effects. We have no serious adverse events. And the side effects are the kind of side effects that you want to see. It's a sign of an immune response. So this phase 2 trial was randomized, multi-center, led by MD Anderson. And we followed these patients for 5 years and had no recurrences in the treated arm. So we're very excited that this year, hopefully, we'll be starting our phase 3 trial. So we have our manufacturing almost completed. We have the GP2 in vials, and we're planning to release the product to the clinical sites. We're doing the release testing right now. We have the protocol already completed and the statistical analysis plan also completed. These are being reviewed. And we have clinical sites that are negotiating with us a budget and negotiating with us a contract so that we can start treating patients before the end of the year. So this is a very exciting time for us at Greenwich. We've completed 1 year since going public. So we hope the second year will be as exciting as the first year was. So we have additional phase 2 trials or phase 3 trials that we can pursue as well. So we can focus on not just HER2 positives, but also HER2 lows, which are 1 to 2 plus. That would dramatically increase the market. In the phase 3 trial, we're actually going to explore other HLA types. So we have a third arm. So without doing an additional trial, we're able to cover that opportunity to expand the market. There are other peptides and checkpoints that we can combine with. And there are also other HER2 cancers that we could pursue. In breast cancer, 75% of the patients express the HER2 protein. In other cancers, it's much lower. But we have really good diagnostic technology to find them. And finally, our ticker is GLSI. We've raised about $36.5 million since our IPO. And as of early September, we were one of the best performing IPOs since early 2019. So this is a summary of all of the data that we published last year. And I'll just go through this in a little bit more detail. I'm going to skip through it faster as we go through the rest of the presentation. So if you can see my mouse, at the San Antonio Breast Cancer Conference on December 9th, we presented this curve at the top, which is 100% disease-free survival or no breast cancer recurrences over 5 years if the patients received their first 6 injections, which gave them their peak immunity. And that's treated arm versus placebo, which is 11%. And that is which is 89% disease-free survival, which is statistically significant. So at the AACR conference, we presented our peak immunity data. And so here you can see in the blue curve, which is GP2 treated patients, we saw peak immunity at about 6 months. And this peak immunity was sustained over time using the boosters. And that's what led to this outcome of not seeing any metastatic breast cancer recur. We also announced at ASCO our safety data. And here you can see that the safety side effects, which are injection site reactions and flu-like symptoms, which are what you see with the coronavirus vaccine. And they're a good thing. They're a sign of an immune response. We saw those during the first 6 injections over the first 6 months, which is what these arrows represent. And then we had boosters every 6 months, so we had 4 boosters. So these 10 injections over 2.5 years led to the safety profile where after the 6 injections, the safety incidences went down, then they came back up. And so this was during the booster period, just like we're thinking of getting boosters for coronavirus vaccine. And then after all the injections were gone, the safety side effects went away. And then we basically completed the trial. So we've had no serious adverse events attributable to GP2 during all the patients that we treated, which is over 4 trials, and that's roughly 138 patients that have received GP2. So the breast cancer market is very large, and the products that we're working in synergy with, Herceptin and Kadcyla, they reduce the recurrence rate 50%. So the patients who are going to recur, half of them respond to these antibodies, but the other half don't. And fortunately, the conclusion of this slide is that we, in combination with Herceptin or Herceptin ADCs like Kadcyla, can bring that recurrence rate down and address the 50% of the patients who don't respond to these antibodies. And if so, we'll be able to provide a complete solution to these patients. In our first indication, we expect to treat about 17,000 new patients per year, which would save about 1,500 to 2,000 lives per year. So this is a timeline, and what you see here on the left here is surgery. So after surgery, you either get Herceptin, Perjeta, or Kadcyla treatment. Then you get, as represented by these arrows, our 6 treatments over the first 6 months where you get your peak immunity. We call that the primary immunization series. And then you get a booster every 6 months. So you get 5 boosters. That gives you a total of 11 doses over 3 years for the phase 3 trial. And from a clinician's perspective, what the clinician first does is diagnose, then they have surgery for the patient, and they give them the anti-HER2 or trastuzumab or Herceptin-based treatment. And then what we're doing is giving them 11 doses over 3 years. And this is a period when the patient is hoping their breast cancer doesn't come back, because if it does, it's generally metastatic and it's deadly. So if we're able to prevent the recurrences during this period of time, we're also going to be making a big dent in metastatic breast cancer. And this is what has created the excitement and the response to our company in December last year. The other thing we think that patients and doctors will do is they'll look at the drugs that are on the market that are less efficacious and have more toxicity and consider not using them and using our product instead. And that's a process that will take place naturally over time after we have approval, and that's called de-escalation. And one of the reasons they're going to de-escalate is because our product in our phase 2 trial was synergistic with Herceptin alone. So we did not see any recurrences, but we did not have Kadcyla, Perjeta, Nerlynx, and 2 new products in HER2 or Tukysa in our trial. So just with Herceptin or Herceptin biosimilars in our product, we have a chance to dramatically reduce breast cancer recurrences. So this is our 5-year data. And if you follow this black arrow, it hits this pink line. This pink line is the recurrence rate for Herceptin alone. And this is old data, and this is where the industry started. So where this black arrow hits is where we start treating. So if we can flatten the curve right where the arrow hits the pink line, the question is, where are we starting? We're starting about 1 year after surgery. We could start earlier. So something we'll do in the future is we'll actually treat patients earlier so we can see if we can stop the first year of recurrences as well. But for this phase 3 trial, we're just going to try and reproduce our phase 2 trial and start where that black arrow is. So just a little bit about GP2. So GP2 is from the intramembrane portion of the HER2 protein. This is the HER2 protein in the cell, so it has an intracellular, extracellular portion, and right here is where GP2 is. And what we're doing is mixing GP2 plus GM-CSF, which is called leukine, in a syringe, and we're injecting it intradermally. We're giving 11 doses over 3 years. So the mechanism is simply when you inject it in the intradermal space, you have antigen-presenting cells that will present GP2, which is this blue rectangle, to T cells. So here in this pink circle is a T cell, and this is the T cell receptor arm. It will bind to GP2 and this HLA complex. And when it binds to it, what ends up happening is we have expansion of those T cells. They're distributed throughout the body. So when this red cell, the cancer cell, comes back, and it naturally presents GP2. GP2 is a peptide that is naturally presented by the cancer cell on the HLA complex. And so these T cells in pink are trained with the TCR binding arm here to kill those cancer cells because we train the immune system to do that. So this is the mechanism by which we can prevent metastatic breast cancer. So historically, we've done 4 trials. So the first phase 1 trial was GP2 plus GM-CSF. In the second trial, we added Herceptin at the same time to evaluate safety, and there were no safety issues. In the third trial, we added another peptide, and we saw no additional safety issues. And this fourth trial is the phase 2 trial. And this is the trial that we reported the data on this past year. So this is the San Antonio Breast Cancer Conference poster that we presented on December 9th. And it's this chart that the public responded to. The HER2 positive arms and the HER2 1 to 2 plus arms were well balanced in the trial. And I'm going to go very fast through these slides. They're on our website under the clinicaltrial.com tab. Sorry, under the clinical trial tab on our website. Okay, so this is the data we showed where we had 0% recurrences over 5 years. We started treating patients about 17 months after surgery, which meant if we follow them 5 years, we actually follow them for 6 years or more after surgery. And what you can see here are these black marks. These are the censoring marks. And what it shows is that in this trial, most of them hover around 5 years. The median is 5 years. And so that's the number of years that we followed these patients. This is the second poster we presented at AACR. It's our immune response data. And I've shown the summary chart before, so I won't go into this in detail, but here you can see our peak immunity at 6 months. And we used 2 assays. We used a binding assay, which measures the quantity of T cells that react to GP2. And we did a skin test, almost like an allergy test, to see if patients react to GP2. They come back 3 days later, and we measure the injection site reaction diameter. And the larger diameter that we see, the more immune response that the patient has against GP2. So this is the dimer data, and this is our DTH data. So in the DTH data, at 6 months, we had 10.8 millimeter diameter reaction in the treated arm versus the placebo, which had a median of 0. And this is our baseline. So we had a nice immune response in the GP2 treated patients, and that's both HER2 3 plus and HER2 1 to 2 plus. If you take this 10.8 and you split it up into the HER2 3 plus, which is 11.5, and the HER2 1 to 2 plus, which is 8.5, we still showed a good immune response in the low HER2s. And this is important for us because it's a sign that we're going to be able to treat the low HER2s as well. At least we're going to get an immune response. And the question is if they have enough cancer cells that have the GP2 molecule presented that will allow us to make a difference in those patients. In those patients, we'll be treating them with Herceptin in the future and with possibly a checkpoint inhibitor. In our phase 2 trial, we did not have Herceptin. It wasn't approved and it's not standard of care, and we did not see a difference in the HER2 lows. This is our safety data that was presented at ASCO. This is the chart I showed you earlier where we combine efficacy, immune response, and safety, which is now a complete data set. We have more data that we expect to present at upcoming conferences, so please stay tuned. On the safety side, this chart summarizes the systemic and injection site reactions. And so we had no serious adverse events, as I mentioned before, attributable to GP2. And here you can look at the types of adverse events by HER2 3 plus versus HER2 1 to 2 plus. And you can look at even just the local injection site reactions. And here you can see we saw these kinds of positive side effects or signs of an immune response in both the HER2 positive and the HER2 lows. We also presented and have been continuing to present the design of our phase 3 trial. So at ASCO, this is the data that we presented. So our plans that we presented. So we're going to screen for HLA-2. We'll have patients in the GP2 arm versus the placebo arm. And the other HLA patients will also screen for, and we'll put them in a third arm. So we're going to be able to look at data in both the HER2 positive patients that are HLA-2 as well as the other HLA types. We've also announced the interim analysis design of our trial, which is 500 patients. We're hoping to look for the midpoint, which will be the mid-level of events. We expect a total of 28 events in the trial, which will lead to about 14 events that will trigger the interim analysis, upon which we hope to file then a BLA for conditional approval. I'm going to go a little faster now. So the manufacturing is straightforward. We have patent life through 2032, and the product is registered as a biologic, so we'll have 12 years data exclusivity in the US. And the market has 4 factors that drive the size of the market. One is HER2 positivity, node positivity, hormone receptor positivity, and HLA type. And if you compare our product to Herceptin, you'll get a good feel for our market. In this red box, we'll initially see about half of the market of Herceptin. And then as we expand into these other indications I described before, we should be able to increase the size of the market and grow the market for Herceptin as well. This slide shows the other products that are on the market. They reduce the recurrence rate 20 to 30%, and they add additional toxicity whereby, as I mentioned before, we've been able to reduce the recurrence rate down to no recurrences, and our product is very safe. In the red box are the other cancers where HER2 expression occurs. So these are other cancers which we can target. This is our management team, which is very experienced. We're committed to taking this product all the way to the market. We have Jay Thompson, who's running the clinical trial, and Chris Fischetti, who's handling our business development in our conversations with big pharma companies, as well as in our conversations with smaller companies, as we look to both out-license GP2 as well as to in-license or acquire smaller products to develop our pipeline. We have about 30 million in cash. We have about 13 million shares total outstanding. We, the management team and directors, own a very big chunk of the company still. As I mentioned, we raised 36.5 million last year. We were added to the Russell 2000. And on December 9th, we had a very large share price increase, roughly 30x, which is one of the highest intraday returns that we're aware of for a non-penny stock. There are many transaction comparables for us that we could talk about. The trading comps are up here at the top. They're straightforward. So there are multi-billion-dollar transactions that have occurred over this past year with Gilead, Merck, Genentech, Seattle Genetics, or Seagen, acquiring or partnering with very large companies for multi-billion-dollar transactions. We hope to accomplish something similar to that. So lastly, our goal is to reproduce our phase 2 trial, to optimize that treatment by treating patients earlier, to using better immune response data, to assess when we should be delivering GP2 to patients, and then to expand the market by going into other markets where we can either treat the low HER2s or the other cancers. We potentially will add a checkpoint inhibitor to the combination of treatments in addition to Herceptin. So lastly, our goal is to reproduce our phase 2 trial and our phase 3 trial. We're very close to starting the trial. We have many other indications that we can pursue. Our ticker is GLSI, and we look forward to continuing to give you updates in the coming months. Thank you very much.
Thank you, Snehal. One quick question. Sorry if I missed it, but was there a start date on that phase 3 trial?
We can't put a start date out there because you have to find the patient, you have to open up the clinical sites, and you have to have the drug at the clinical site. So we're working on all 3 in parallel, and that's all we're doing every day. So no start date.
Can you give like an approximation, like a target, like a quarter, Q1, Q2,
well, so
Q3?
We have publicly stated our goal is to start the trial in the 4th quarter.
Okay, great. Thank
you.
you.
So if it's not to treat the first patient, it's at least to enroll the sites and have the sites active, looking for patients.
Got it. All right, Snehal Patel, Greenwich LifeSciences. He's the CEO. Snehal, there's some pretty encouraging data there. Good luck on the next trial, and thanks for coming on the Small Cap Conference today.
Thank you very much for having us.